Okay. So, of course, the word immunodeficiency is just direct as it is. This is a situation where the immune system is unable to fight infections because either absent or it is there but in very low levels. It is reduced, you can't really have it. So we have scenarios, we have reasons as to why the immune system can behave that way. It would depend maybe with the host. The host can have an impaired immune system, either because some type of cells of the immune system have been affected. For instance, we've seen that we have two types of immunities, the innate and adaptive. We've seen that the innate immune system is run by quite a number of things. We've also seen that the adaptive immune system is run by quite a number of things. We saw that inflammation plays a key role. We saw that phagocytosis plays a key role in all this. We've also seen that cytotoxicity plays a key role in all the two wing arms of immunity. We've seen that the adaptive immune system is run by three types of cells, specifically T cells, B cells, and antigen-presenting cells. Those are cells that run the adaptive immune system. So you can find scenarios where either naturally, which means at birth, or acquired later on, the body is unable to produce enough of this. And that, of course, then triggers a scenario of immunodeficiency. So that we can divide immunodeficiency into two types. We have primary immunodeficiency, and this is congenital. It's present at birth. This especially due to genetic defects. Then we have secondary or acquired immunodeficiency. This develops later in life due to external factors. And this could be things like HIV. We know that HIV is the main cause of immunodeficiency in our populations currently. Other reasons would be things like malnutrition, of course, chemotherapy, immunosuppressive drugs. If somebody maybe is taking immunosuppressive drugs, then definitely they are going to have a scenario of acquired immunodeficiency. But we also have the main part where we're saying that there is primary immunodeficiency, is congenital, is there due to defects in the genetic system. So we're going to start with the primary immunodeficiency and see what are some of the conditions that exist that are referred to as primary immunodeficiency. And if they are there, and of course we are saying that they are genetic defects, what are some of these genetic defects that would cause an individual to have a primary immunodeficiency? And what do we call a primary immunodeficiency? So of course they are congenital, we already said. They are not caused by environmental factors. They are not acquired, okay? They could be autosomal or X-linked disorders. Now, when you talk about autosomal, these conditions are carried on either chromosome number 1 to chromosome number 22. Those are referred to as autosomal chromosomes, or the X chromosome. We know that for male, an individual that we describe as male, they are going to have 46 chromosomes in somatic cells, or 23 chromosomes in their gametes. For male, of course, in the sperm. For female, in the ova, they are going to have 23 chromosomes. In their somatic cells, they are going to have 46 chromosomes. And these 46 chromosomes are a pair, of course, each chromosome being given from their either parent. A mother donates one and father gives the other. So if you have 46 chromosomes, you've inherited one chromosome from your father, one chromosome from your mother. Then if you're male, you inherited the X chromosome from your mother and a Y chromosome from your father. If you're female, you've inherited X chromosome from your father, X chromosome from your mother. That is how we define, of course, the genders. Now, when you say a condition is autosomal, this condition could be happening in any of the genes that is from gene number, I mean not gene, but chromosomes, chromosome number 1 to chromosome number 22. Then it's also possible to have these disorders being carried on the X chromosome. We are not talking about the Y chromosome here because it's not been implicated in any primary immunodeficiency so far. We have different aspects that could be affected. Issues to do with complement. We saw what complement is and mechanisms for which complement is activated. So we saw that complement is a group of proteins. These proteins run a cascade, and being proteins, they are definitely for by a certain gene. If they are being coded for by a certain gene, that gene is carried on a chromosome. Of course, chromosomes carry DNA. So complement has its group of genes. In case of a genetic defect where the complement is not being produced enough of it, we saw how important complement is. We saw that complement is a link between adaptive and innate immunity. We saw that complement would act as an opsonin. We saw that complement will produce a mark, the to eliminate pathogens. And so it's a very, very important protein. If it is not being produced in enough of it because of, say, mutations in genes that code for complement, then definitely these individuals are going to suffer from a primary immunodeficiency of some sort. Antibodies, we know that antibodies are of course, they are gene products. We saw that we have what we call a super gene family, globulins. They produce bodies, IgM, IgG, IgA, IgD, IgE. We all saw that. And these genes, if they have issues, they have mutations, and antibodies are not being generated, then it means the individual will not neutralize antigens that are infecting. Similar to phagocytic cells. Phagocytic cells, the three main phagocytic cells, two main phagocytic cells being neutrophils and macrophages. If there are issues to do with how these cells are activated, maybe interferon gamma is not being generated enough of it so that then it's able to activate macrophages, so that it's not able to enable neutrophils from proliferating, then definitely individuals are going to suffer from multiple infections, and that is primary. Another key aspect, of course, is the T cells. If T cells are not being generated in enough of T cells, then definitely the immune system will be compromised totally, because you've seen that T cells play a very, very key role. They are the ones that run the cell-mediated immunity. For you to realize any cell eliminating the causative agents, pathogens, then you need T cells, of course, for that to happen. So they are quite important. And in case they are deficient, then of course individuals are going to suffer multiple infections. They are going to suffer from even non-communicable diseases such as cancer cannot be eliminated by the immune system if T cells are deficient. So it's not just infectious agents only, even non-infectious agents. Autoimmunity, of course, will be pronounced in case there's such a problem. So let us look at these deficiencies of that cause primary immunodeficiency systematically. Let's look at them systematically, starting with issues to do with phagocytic cells. Issues to do with phagocytic cells. Now, in case there are phagocytic cell defects, in case there is phagocytic cell defects, we have a condition that has been described called chronic granulomatous disease. Chronic granulomatous disease. Now, I've just described something called a granuloma. I've described something called a granuloma. Can anybody remind us what we mean by a granuloma? I've said what a granuloma is. Yeah, so Steve has answered correctly. It's giant, not macrophages, but macrophage, giant macrophage. So these are merged cells. So you're picking macrophages, and group. So it's like you're merging cells. You're taking one cell and another cell and another cell and another cell. Of course, we know that macrophages are resident in tissues. So you're picking these macrophages with the help, of course, of interleukin 4. These cells are able then, for example, yeah, I'm seeing Clinton giving a very good example here, giant macrophages that trap egg cells. Yeah, I also gave another example that we are most likely we have latent TB, and we have latent TB because the TB has been trapped by macrophages. And these macrophages, these giant cells are the ones we are referring to as granulomas, and they are really require interleukin 4. Okay, so this is called chronic granulomatous disease. And we know that macrophages emanate from monocytes. So this is a rare disorder where monocytes and neutrophils cannot produce reactive oxygen intermediates for killing phagocytosed organisms. That's the thing. Macrophages, of course, which are mature monocytes, as well as neutrophils, kill mostly by producing reactive oxygen intermediates that we saw. And so, if that is the case, these are individuals born with cells that are unable to produce reactive oxygen intermediates. And therefore, they are going to suffer quite a number of infections because most of the infectious agents require elimination by reactive oxygen intermediates. We saw that. We saw how the reactive intermediate oxygen intermediates interfere with the membranes of these infectious agents and lead to their death. So these individuals are going to have recurrent bacterial and fungal infections, and they are often resulting in granulomas. So because these cells are unable to work out well, they are probably going to merge and form granulomas. These cells are going to merge and form granulomas. So of course, these individuals mostly suffer from Staphylococcus aureus. These are bacteria, of course. That they are going to suffer from fungal infections like Aspergillus fumigatus, Candida albicans. They are going to suffer from that. We try to treat these individuals by, of course, you induce this by interferon gamma can be used in their treatment. I don't want to go into the issues of treating them. What is important for us is to know that for individuals who have chronic granulomatous disease or CGD, they have monocytes and neutrophils, but these monocytes and neutrophils cannot produce what? Reactive oxygen intermediates. That is it. So of course, these are phagocytic cells. Monocytes by themselves are not phagocytic cells. They have to mature to become macrophages for them to become purely phagocytic. So we're just talking about the same thing. Then we have this condition called the Chidiac-Higashi disease. The Chidiac-Higashi disease. This is a rare autosomal recessive disorder, and this results from mutations in the CHS gene. Okay, so it's the good thing about these conditions, they are rare, the two of them, they are rare, but it's autosomal recessive. Could somebody remind us what we mean by a recessive condition? Autosomal, we know that is either being carried on chromosome either 1 to 22, one of those chromosomes. That is where the mutation is. So the CHS gene is carried on one of those chromosomes. Let me just confirm. CHS gene is on chromosome. Which chromosome carries CHS gene? What's a recessive condition? So it's a multi-gene. CHS gene is a multi-gene. It's not just found on one chromosome. It's found on chromosome 2, chromosome number 7, as well as chromosome number 9. So it's on a number of chromosomes because it's just like the superfamily gene of immunoglobulins. Yeah, so what do you mean by a recessive condition? Anyone? A recessive condition. You're either having a recessive condition or a dominant. What do you mean by recessive?
So a recessive condition is the one that only expresses itself when it's in a homozygous state.
Okay, that's a good addition. So, which means you need two alleles. You need two alleles of this particular gene to then see the expression of the phenotype. In other words, as we are saying, this gene is carried on chromosome 2, 7, and 9. So if you're inheriting, let's say the one that is on chromosome 2 is the one that is being expressed here. If you're carried, if it's being, the mutation is carried on chromosome 2 of your father, then your mother should also carry the same mutation for that to be expressed in homozygous state. But if it's heterozygous, maybe your father doesn't have and your mother does, and so you have a scenario whereby you are heterozygous, it will not manifest. So it requires, it's just like sickle cell, it's a recessive condition. Both parents have to be carriers to give birth to an offspring, and that offspring has to inherit both. So both the mutant alleles for then expression to occur. If that doesn't happen, then they are not going to phenotypically express with that condition. So that is what we mean. So it is a recessive disorder, mutations in the CHS gene. This is characterized by giant lysosomal granules, the phagocytes, and this leads to defects in chemotaxis, phagocytosis, as well as microbicidal activities. So these individuals end up having giant lysosomes. And this, we've seen how lysosomes are important. They play a key role, of course, in enabling defenses, especially phagocytically, as well as just eliminating any causative agents. They're also able to break down proteins from the invader. So when they are giant, they have nothing. And when they have nothing, they can't perform the roles they are supposed to. Then we have the leukocyte adhesion deficiency. Leukocyte adhesion deficiency. This is caused by lack of CD18 beta subunit of the beta integrins. Okay. So of course, this adhesion, well, it means it needs to attach to another cell, maybe to trigger a cellular response. So individuals, of course, will end up having pyogenic infections. These are infections that especially are characterized by pus. Infections characterized by pus. And of course, they have problems in wound healing. Then another condition that is phagocytic related is defects in interferon gamma interleukin 12 axis. Interferon gamma interleukin 12 axis. Here, patients have mutations in the interferon gamma receptor as well as the interleukin 12 receptor, or interleukin 12 itself is not properly secreted. So, of course, they also have, they are unable to deal with mycobacteria. They are unable to deal with, as well as other intracellular bacteria. So these individuals, of course, are unable to have an interaction between interferon gamma as well as interleukin 12. It's like this interferon gamma usually interact with each other. They're able to interact with each other. And if this doesn't happen, then of course these individuals cannot deal well with bacteria. Okay. Then we have primary immunodeficiency complement system deficiencies. Due to the complement system deficiencies, if you remember, the complement system is activated via, we have three pathways. If you remember, we had, of course, the mannose binding lectin pathway. So we also have the classical pathway as one of the other pathways. And then what is the other pathway? We have this other pathway that was dealing with MASP1 and MASP2. So we have those three pathways that, of course, activate complement. So, mannose binding lectin deficiency. Mannose binding lectin deficiency. This mannose is present, of course, on bacteria in most cases, or the causative agent, but it needs its receptor. And if its receptor is not present on the cells, then of course these individuals will not detect any bacteria that have mannose on them or other carbohydrates on them, and therefore they are prone to having infections from bacteria that carry the mannose, especially on their cells. Early classical protein pathway deficiency. These are susceptible to immunocomplex diseases, and individuals, especially systemic lupus erythematosus, are likely to have this deficiency. The classical pathway. Remember the classical pathway ndio ile where there is just usually normal level of C3 being broken down, then this C3 again inakuwa activated by C4 is broken down to C4B and C4A. Of course, C4B binds to C3B and it becomes a convertase of C3, and C3 again is broken down to C3B, and then of course binds to C5B and becomes a C5 convertase, the classical pathway. Then C3 or regulatory protein deficiency, we saw that C3 is the most abundant complement protein. And if it's deficient, then of course, users are going to have recurrent infections, especially due to defective opsonization. We know what opsonization is, as well as chemotaxis. We also have individuals who have deficiencies between C5 and C8. We saw once C5 is broken into C5B, C6, C7, C8, and C9 bind to C5B to form MAC. So if an individual is going to have C5 to C8 deficiencies, then they are definitely going to be susceptible to very many infections, including the infections of bacteria that are in the genus Neisseria. Bacteria like the ones that cause what? Neisseria, gonorrhea. The gonorrhea bacteria that causes gonorrhea, of course, is in the Neisseria family. They are very susceptible to it. We have C1 inhibitor deficiency. We saw that C1 was a complement that then can be broken down into C1Q, C1S, C1R. That would trigger the classical pathway ultimately. So if there is inhibition deficiency, then it causes angioedema. And of course, there's always production of vasoactive C2 fragment. We saw that C2 is also plays a key role, especially when you're producing C3 convertase, plays a key role. Then we have GPI anchor protein deficiencies. These are mutations in PIGA gene. Individuals end up having paroxysmal nocturnal hemoglobinuria. So these individuals are able to urinate blood, they generate blood, or rather, the proteins in their urine are it's a dangerous condition also, resulting from, of course, GPI anchor protein deficiency, especially in the activation of complement. They play a key role in complement activation. So those are deficiencies that are resulting from complement issues. Then we have immunodeficiencies that are emanating from antibody deficiency. Immunodeficiencies emanating from or immunoglobulin deficiencies. So we have like the X-linked Agammaglobulinemia, or XLA, sometimes it's called that. And this is due to mutations in the proton tyrosine kinase gene. So these individuals have depressed immunoglobulin production. And once you are unable to produce very little immunoglobulins or antibodies, we know that you are prone to infections. Then we have immunoglobulin A deficiency. We know what immunoglobulin A is, IgA. We've seen that it's present especially in mucosal surfaces. So of course, individuals who have this deficiency will have infection so much, especially that play a key role in the mucosal surfaces. Then we have common variable immunodeficiency, or CVID. These are quite a number of heterogeneous diseases. They could be autoimmune, and these individuals don't have proper T cell signaling. And if they don't have proper T cell signaling, then of course it affects how the B cells are going to be differentiated. And if B cells are affected in how they are being differentiated, then of course it affects how antibodies are going to be produced. Then we have X-linked hyper-IgM. These individuals have very low IgG, IgA, and IgE, but normal high levels of IgM. In other words, class switching is not happening for these individuals. And class switching, of course, requires CD40. This means there is a mutation in CD40 ligand. And if CD40 ligand has been affected, then class switching will not happen. So these individuals mostly end up having IgM. So they can deal with general infections, but specific infections, as infections evolve into specificity, then it doesn't happen. There's also transient hypogammaglobulinemia, that is in infancy. This one is characterized by delayed synthesis of immunoglobulins. Individuals are born with it, some, but it's transient, it's not permanent, it's transient. So usually by age 4, individuals are able to be okay. Okay, if you're born with transient hypogammaglobulinemia, by the time you reach to age 4, it should be able to resolve itself. But these individuals have delayed immunoglobulin synthesis. So they are going maybe to depend on their maternal antibodies quite some time to protect themselves. And once the maternal antibodies start fading, then they might have problems, but hopefully by age 4, they are able now to start producing their own immunoglobulins. Then we have those that are affecting the T cells. Those that are affecting the T cells, we have DiGeorge syndrome. DiGeorge syndrome is the failure of the thymus, leading to development of T cells, and therefore these individuals will have issues with their T cell maturation and T cell production, called the DiGeorge syndrome, and it's as a result of a deletion in chromosome number 22, region 1, subregion 1, band 2. This deletion causes the DiGeorge syndrome. We also have other deficiencies. These are, of course, related to the T cells. We have the Omenn syndrome, and Omenn syndrome is defectivity in VDJ recombination. Somebody was asking me in the previous class that how is it that class switching happens? And I said we have an unrecombinant homologous recombination. We have non-homologous and homologous recombination that happens. So we have what we call the VDJ regions of those genes that then undergo class. So this recombination that happens in the VDJ is what leads to the mutations that ultimately lead to switching. So individuals with Omenn syndrome have defects in that particular recombination, which means class switching is impossible for them. them. We have the bias lymphocyte syndrome. These ones have problems with MHC class 2. And we saw that MHC class 2, MHC class 2 plays a role. What T cells interact with MHC class 2? What T cells interact with the MHC class 2? Somebody remember?
Before.
Yes, especially for extracellular. All right, we have ataxia, telangiectasia. Of course, this one's because of defective DNA repair, especially when DNA is subjected to UV light, it forms thymine dimers, and it's unable to be repaired. These individuals end up with this, and they are unable to produce proper T cells. Hyper IgM syndrome, we already talked about this. They have issues to do with the CDL40, and so CD40L, CD40 ligand. So, and this was important, especially in class switching, as we saw, which means T cells are not interacting properly with B cells, and therefore, as they interact, even as IgM is being produced, it can switch. Then you have severe T cell deficiencies due to defects in CD3, epsilon, CD3, gamma, and ZAP70. These are very key molecules on T cells. These are very key molecules on T cells, and these molecules interact, of course, as they are important in how T cells proliferate. If T cell proliferation is affected, then of course mutations in these regions, CD3 epsilon, CD3 gamma, and ZAP70. These are proteins on the surface of CD3s, that is T cells. So of course, then that of course interferes with the proliferation of those cells. Then we have SCID. SCID is the most common primary immunodeficiency, but we have different forms of SCID. SCID stands for severe combined immunodeficiency. It's a combination of several factors. It's not just one factor like we've seen. We have phagocytic defects, we've seen immunoglobulin defects, we've seen T cell defects. SCID is a combination of several issues. It's a combination of several issues, not just complement, not just T cells, not just immunoglobulins, not just phagocytic cells. It's a group of diseases involving T cell deficiency with or without B cell or natural killer cell deficiency. It's quite a number of things that are involved here. We have SCID, where we have mutations in the common Y chain of interleukin. We've seen the ones that and this is going, and if they are going to produce interleukins, their gamma chain is important. We also have autosomal recessive SCID due to defects in purine metabolism. Then we have other variants of, which is ones caused by RAG gene mutations that is affecting the VDJ recombination. We also have mutations in the kinase. These are just pathways that activate the immune system, like the JAK pathway. It activates the system, especially in proliferation of T cells and interactions of T cells to B cells. So again, if that pathway is affected, of course, that pathway is made up of several enzymes. If enzymes like the JAK kinase is interfered, the gene that codes for it undergoes mutation, then of course that pathway is also interfered. So those are SCID. We have WAS. WAS it's another form of so it's not SCID, but it's just an immunodeficiency that combines quite often a defect, and it's an X-linked disorder, which means it's carried on X chromosome. So the Wiskott-Aldrich syndrome, and these individuals end up having thrombocytopenia, eczema, and recurrent mutations, recurrent infections. Sorry, this is due to mutations in the WASP protein. Mutations in the WASP protein, that is the Wiskott-Aldrich syndrome protein. Mutations there in that particular gene then lead to those conditions. We've already talked about ataxia, telangiectasia, autosomal recessive, due to issues to do with DNA repair. We have X-linked lymphoproliferative disease. This one is associated with Epstein-Barr virus infection, the EBV. The Epstein-Barr virus will introduce mutations, and once it introduces mutations, then the way in which of course, these mutations are being introduced in the SAP protein, the gene that codes for SAP protein, and then it leads to issues to do with proliferation of lymphocytes. So lymphocytes are not proliferating properly. So how you're able to identify these issues is by measuring immunoglobulin levels and antibody responses to vaccine. So if you want to see if, say, your immunoglobulins have issues, you measure levels. We have normal levels, normal titers that we expect if somebody either has been infected or if somebody has been vaccinated. There are certain levels of immunoglobulins or antibodies that need to be generated in the body. So if they are below par, then you know that there's an issue with this individual. You can also count B cells using flow cytometry, and B cells are referred to as CD19. B cells also carry CD20 and CD22 on their markers. So you can be able to count them using the flow cytometers. You'll know whether there is an infection or not. T cell elimination, you're able to count T cells also, and T cells are CD3s. They have other surface markers such as CD2, CD5, CD7, of course, CD4 and CD8. These are the most common ones. So you can be able to measure them using flow cytometry, of course. It's a very, very good technique of counting cells, and you're able to tell whether there's an infection. Several tests can be done, like the baculin test. I'm sure most of us who have undergone BCG, you have some marker on their skin. So of course, that would be the tuberculin skin test. Just shows you that your immune system is okay. But you can test them against Candida, and of course, you can trigger cytokine production assays to be able to see whether your T cells are functional or not. Phagocytic functions, we can also do laboratory experiments and see whether these phagocytes are able actually to work properly. In vitro tests, we can do them to see whether the phagocytes that are being generated by the body, are they able to do their phagocytic functions? Are they able to interact with complement, for instance? And once complement creates an opsonin, are they able then to phagocytose it? So that is how you're able to detect if an individual has problems with the immune system or not, especially for these primary ones. For the acquired, of course, causative agents can be known, but for primary ones, they are genetic conditions. So you can only do a test for you to be able to know whether an individual has issues or not. But of course, you're able to tell there's a problem if they are suffering from recurrent infections. If any infection is able to bring them down all the time, then definitely these individuals have issues to do with their immune system. Okay, so that is it for today. I think in the next lecture, we'll be looking at acquired, and for the acquired, we're mainly going to focus on HIV. There are other reasons as to why people are able to acquire infectious, secondary or acquired immune deficiencies. It's possible for other reasons we've seen, but we are going to mainly focus on HIV being the main one that causes secondary immune deficiency or secondary immunodeficiency. But these ones are primary. What we've talked about majorly are primary causes, the ones especially that are genetic. And we've seen they could either be phagocytic, the ones that are affecting phagocytic cells. They could be the ones that are affecting complement. They could be affecting B cells or rather immunoglobulins in general. They could be affecting T cells. It could also be a combination of several factors, what we are referring to as SCID. And it could also be SCID SCID majorly deals with T cells, and then we have other combinations, of course, of several reasons as to why then an individual is going to suffer from immunodeficiency, like the ataxia, like the X-linked lymphoproliferative disease. These are several combinations of factors, of course. Yeah, so let's stop there for today.